Hello, I'm Russell Schwartz from Carnegie Mellon University, and I'll be presenting work of my co-authors and I on data science training for experimental biology graduate students, and in particular on our efforts to provide this training for a cohort of primarily experimental biology doctoral students. It's now been recognized for decades that life sciences research and practice has become critically dependent on bioinformatics and on related topics in mathematics, statistics, and computing. Because of this, education of future life scientists needs to be reformed to prepare them to work in a field of quantitative and data-driven biology. For example, I highlight here a recommendation from the U.S. National Academy of Sciences' influential Bio 2010 report calling for such curricular reform. For anyone unfamiliar with Bio 2010, the report actually came out in 2003, and the name was meant to be aspirational for where biology education needed to be by 2010. Suffice it to say, 2010 has come and gone. It's now 2020, and yet progress toward universal, rigorous bioinformatics education of life sciences students has been minimal. As a more recent study by Jason Williams et al. Highlighted, most undergraduate programs in biology, at least in the US, still essentially cover the same content they did 20 years ago and provide no appreciable training in bioinformatics. Programs offer many reasons for this failure. They lack qualified faculty to teach it, for example. Students don't want to take it. They can't fit it in their curriculum. But the bottom line is that progress has been glacial while the need for it has grown steadily more urgent. This talk is about an effort to figure out how to better meet this need for one cohort. I believe that we can often best help drive systematic reform by figuring out what works in our own institutions and exporting our successful strategies. And this is a topic where we felt Carnegie Mellon was in a good position to aim for a higher standard. We have a long history of offering training for computational biologists, so that is for real interdisciplinary specialists at the bachelor's, master's, and PhD levels. And so we do have a lot of experience teaching interdisciplinary classes in the area and many qualified faculty members to teach them. Bringing appropriate computational training to every biology student is a harder challenge, though. Raising our standard at the undergraduate level is a work of many years. We're unusual in that we've actually required all of our bachelor students in biology to take computer programming since 1995, and we did push through a requirement for a full computational biology class required of every biology major since 2010. Most recently, we added a requirement for statistics by making this a prerequisite for our required genetics core class, and so we think we've reached a point where we're providing good training to our undergraduates in what they need to know about computational and data science work. The PhD level, though, has proved to be a much more difficult challenge, and that's what I want to talk about today. These are students for whom we have few course requirements in general, since we want them to get past their classes early and focus on research. Furthermore, the field as a whole has no expectation of bioinformatics training at the bachelor's level, and so most of our students are coming to us with no prior training in this area. Yet we have a near universal recognition from our faculty that these students need skills in data analysis to work in a modern research laboratory. And so we set out to design a class to serve the needs of this cohort. Our goal was to provide training that would prepare students for diverse paths and thus cover many different experimental modalities and different biological problem domains. We needed this training to be suited to students who would be designing and conducting their own experiments, and so placed a heavy emphasis not just on how to analyze data handed to these students, but also how their knowledge of data science needs to inform their experimental design practice. Furthermore, we were looking not just for plug-and-play training, that is, plug in this kind of data into this box and push that button to do a particular analysis, but training that would provide students enough grasp of the fundamentals that they could generalize this knowledge and adapt it to new situations, new data types, and new problem domains. Furthermore, we had to do that given the assumption that our students usually have no prior training at the undergraduate level, and so we can assume no prerequisites, and that they can't take large numbers of courses or multiple semesters to reach competency, that we have a limited amount of time, effectively a single one-semester course, to provide them all of the training they need to start working productively in research labs. We captured these goals in a set of learning objectives, which I show here briefly, but I'm not going to go through in detail. These essentially formalize the ideas laid out in the previous slide. We then developed a course to meet that need, which has been through 3 iterations now. We first piloted a stripped-down version of the course based around 3 topical biological modules, and combining lecture material and hands-on workshop sessions analyzing data in class using R with RStudio. In 2 subsequent iterations, we sought to improve on that course by expanding the topics covered. We increased our coverage of other areas of biology as well as bioinformatics and biostatistics. And we added supplementary instruction with a resource from Carnegie Mellon's Open Learning Initiative, or OLI, to provide offline self-paced training in essential background material for the class. Finally, we switched from RStudio to Jupyter Notebooks as our primary platform for hands-on in-class instruction to address difficulties we had run into in earlier iterations. I'll say more about both of these kinds of technology-assisted learning mechanisms shortly. We converged on a course structure that in its latest iteration is shown here. The course is organized around a set of modules, each introducing a different biological domain and data type, with lectures introducing the biology and the biotechnology behind that particular topic. These biology and biotechnology lectures are taught by co-instructor domain experts who work in this particular problem domain and are familiar with the tools and analysis that we'll be talking about there. Lectures also provide coverage of computational concepts that we encounter in these different modules, each typically introduced in one module and then reinforced in the subsequent ones to help students understand how common computational and mathematical abstractions can apply across different biological contexts. Each module involves a mix of these lecture sessions with computer lab workshops where we sit down with real primary data from our co-instructors or from other laboratories in the department and work through hands-on analyses involving the concepts presented in the lectures. In covering computational and statistical material, our goal is to provide enough of the foundational concepts that students can see how the same ideas apply across domains. For example, we look at the intuition behind a t-test in the context of differential gene expression analysis and see how to use that statistical test to identify differentially expressed genes. But we also examine how we can use an intuitive understanding of that statistic to help guide experimental design in that domain. We then later see how a mathematically similar problem in a different domain, quantitative trait locus analysis, can lead us back to the same tool for a seemingly very different biological problem. This approach is repeated for a variety of topics such as clustering, classification, and dimensionality reduction. So, for example, we can introduce a concept like clustering in one context. In this case, looking particular at an application of clustering in developmental biology, working with RNA-seq data generated from a gene knockdown experiment to try to understand modules of genes that are controlled by the gene that was knocked down. And we can then switch to a different biological context and see how the same concept of clustering can address a mathematically very similar question in a quite distinct problem domain, in this case identifying neurons acting in concert in performance of a task by a mouse from single-cell electrophysiology data. We introduced Jupyter Notebooks in our latest iteration to provide a smoother and more reliable interactive experience than we had had trying to work in class with RStudio. For anyone unfamiliar with Jupyter Notebooks, they're effectively a way to combine annotation and code in a single document that lets students work through a computational workflow interactively. It lets us provide a kind of script with code snippets that students can run on their own computers, but that they can also edit or add to so they can see how changing the code or changing the data that's analyzed in it changes their results. This allows us to provide the students incomplete or buggy code, for example, and let them reason about how to fix it or explore effects of possible changes in the code on their analysis. We also found it valuable to introduce material from Carnegie Mellon's Open Learning Initiative, or OLI, to address challenges of trying to serve a cohort with diverse levels of background training in statistics. OLI develops a kind of intelligent, interactive textbook to allow students to learn at their own pace. This combines didactic instructional material, automated interactive exercises, which provide automated guidance to students as they proceed through them, and integrated assessments let instructors track the students' progress offline. We found this tool a very effective way to bring students up to a common level of background knowledge without unnecessarily burdening those with more prior preparation. We felt at this point that we had put together a course that should be serving the intended learning objectives. We have to assess that though, and so did pre and post testing to see if students were developing the mastery we intended. While the sample size is far too small to draw definitive conclusions, we found it at least suggestive of where the class is and isn't working well. To illustrate what the testing looked like, I'm showing here a sample question from a pretest. This question describes an RNA-seq experiment and asks students to say how they might change the experimental design to improve the study's power to detect weaker effects, a topic intended to draw on their understanding of both the statistical tools and the biotechnology and biology that question involved. Again, our sample size was too small to have significant results, in part because this is not a large cohort, and in part because some students joined the class late and missed the pretest. Nonetheless, the answers are at least suggestive of where the material is or isn't meeting our learning objectives. We observed that students did improve, although only slightly, in their ability to provide correct answers to these kinds of free answer assessment questions. And we also found that there was a somewhat more substantial reduction in incorrect answers. So this does suggest that the students are seeing some improvement from the beginning of the class to the end. The results were not as good as we might have hoped, and one possible reason is suggested by breaking down the performance to distinguish answers relying on quantitative knowledge versus those relying on biological knowledge. Students showed notable improvement in their post-class testing in offering correct answers that drew on the quantitative concepts we teach in this class, but they actually end up making less use of their biological knowledge, even when that biological knowledge is actually appropriate for answering the question. This suggests that we might need to better focus on how students should integrate these domains of expertise in their reasoning. And that, I think, allows us to draw some conclusions. We've created a course to serve what we see as an important and still largely unmet need for data science training for experimental biology students. Key elements of the course include the use of primary research data and domain expert co-instructors, a focus on concepts rather than tools with reinforcement across disciplines and data types, and a combination of traditional lectures with interactive computer lab workshops and automated offline technology-assisted learning resources to better customize the student experience. Assessment cannot yet prove the course is effective, but it does at least suggest modest improvements in ability to give correct answers and avoid incorrect answers, as well as students feeling more comfortable in relying on their quantitative knowledge and skills, even if they are perhaps drawing less than they should on their biological knowledge post-course. There's still a lot to do in the future to improve this course. We would like to see if we can strengthen performance in the assessments I just mentioned, for example, by focusing more on the issue of better integrating biological and computational domain expertise. We hope to continue expanding our course modules, in part with the goal of creating more easily exportable content for other classes at CMU and externally. And we also want to see if we can improve coverage of more challenging topics. Informally, the biggest complaint we hear from students is that the course doesn't go far enough. They want more advanced advanced skills and especially want to learn how to write more complex computer programs, something we simply didn't have time to cover in this class. While we were disappointed not to be fully meeting the student need on this point, I will say that we're heartened that the students want more advanced training than we were able to give them. This is a marked contrast to my own experience developing similar material for biology undergraduates, as well as to data from the Williams et al. Paper I mentioned at the beginning of this talk, which found that many biology undergraduates resented having to take bioinformatics and related quantitative material. Our experience is that our graduate students, even ones who had no prior bioinformatics training at the undergraduate level, know they need this material and are willing to work hard to acquire it, and it's just up to us to figure out how to deliver it to them. I'd like to close with some acknowledgements. First of my co-instructors, Veronica Hinman, Sandy Coleman, Louisa Hiller, and Allison Barth, as well as other department members, Eric Itchery, who provided additional data for the analysis, and Aaron Mitchell, who was the department head when we first offered this course and was very supportive and helpful in driving it and making it possible for us to implement this. I'd like to thank the department for supporting our efforts in this, the Eberly Center for Teaching Excellence and Educational Innovation at Carnegie Mellon for their assistance with our assessment strategies, and the Open Learning Initiative for help with incorporating their automated teaching tools into our work. And thanks to all of you for your attention.